we hear so much about cancer treatments, like chemo and radiation. They kind of just attack everything. And yeah, they can work, but they also hit healthy cells too, right? It makes you wonder, what if we could be more precise?
Yeah.
What if we could target just the cancer cells? And even more specifically, what if we could go after the cancers that have like these underlying DNA repair problems? Because we know those exist.
Oh, absolutely.
So that's kind of what we're diving deep into today. It's this really exciting area of research.
It's huge. I mean, think about it for a lot of cancers, like some breast and ovarian cancers, and even these aggressive brain tumors called glioblastomas. The root of the problem is that they can't fix their DNA properly.
Yeah.
And that's a vulnerability for sure, but it also means they've got to rely on other ways to survive.
Exactly. And that brings us to this fascinating new study out of the University of Iowa. It was just published in Nature.
Oh, wow.
And they had some researchers in Italy that were really key to this too. What they did was they kind of like peeled back the layers of this protein called RAD52. And what they found is pretty groundbreaking.
Yeah.
And they used some really high-tech tools to do it.
I was going to say, what kind of tools are we talking about?
Oh, things like cryogenic electron microscopy.
Wow.
It's like they froze a molecule in action and then took a super detailed picture of it.
That's incredible.
I know, it's crazy, right? So our goal today is to understand this surprising structure of RAD52, because it could mean a lot for, everyone listening. It could lead to cancer treatments that are not only more effective, but maybe also have fewer side effects.
So much gentler on the body.
Yeah, exactly.
Yeah.
So maybe let's just start with the basics. You mentioned cancers that have problems with DNA repair.
Yeah.
Why is that such a big deal? Like, why is RAD52 so important to study in those cases?
Okay, so think of your DNA as like a really detailed blueprint for your cells, right? And it's always facing these little accidents, like damage from all sorts of things.
Just normal wear and tear.
Yeah, exactly. Now, usually there's this whole team of repair workers inside the cell that fix those errors super quick.
Right, like a cleanup crew.
Exactly. But in some cancers, especially the ones with defects in these genes, we hear about a lot, like BRCA1 and BRCA2.
Yeah, those are the ones often linked to breast and ovarian cancer.
Right. Those primary repair teams are basically out of commission.
Oh, wow. So they're really in trouble.
Yeah, but the thing is, these cancer cells are still dividing like crazy. They're trying to grow, so they desperately need a backup plan.
Makes sense.
Yeah.
So that's where RAD52 comes in.
Exactly.
Yeah.
It's like the star player on that backup DNA repair team.
So even with all this DNA damage, these cancer cells can still survive because of RAD52.
A lot of times, yeah. They're basically leaning on it hard. And that's what makes it such an interesting target for therapy.
Okay, I see what you're saying.
Yeah.
So if we could block RAD52 in these specific cancer cells, the ones that are already struggling with DNA repair, then they would basically self-destruct.
Yeah, they'd be overwhelmed by their own genetic damage. It'd be like cutting off their lifeline.
That's a powerful image. And, this whole idea of targeting these backup mechanisms isn't totally new, right?
Right.
We've seen this with PRP inhibitors.
Oh, yeah, absolutely.
So where does RAD52 fit into all of that?
That's a great question. PRP inhibitors were a big deal for treating DNA repair-deficient cancers, especially the ones with BRCA1 or BRCA2 mutations. PRP is another protein involved in DNA repair, and blocking it in these cancers can be really effective, at least at first.
At first, what do you mean?
Well, some patients have these amazing periods of remission, but the problem is that cancer cells are sneaky.
Oh, boy.
They figure out ways to resist the PRP inhibitors over time. They adapt.
So that's why we need more options.
Exactly. We need to find other key proteins in these alternative repair pathways, like RAD52.
Okay, that makes sense.
Yeah.
So maybe hitting RAD52 by itself or even combining it with PRP inhibitors could help overcome that resistance.
Possibly, yeah. And it could help more patients, too.
So RAD52 is this promising target. We've established that. But how do we actually design drugs to go after it?
Yeah, that's the million-dollar question.
And that's where this new research comes in, right? This study that looked at the structure of RAD52.
Exactly. We need to understand at a fundamental level, and what they found was really unexpected.
Okay, I'm intrigued. What did they discover?
So they used cryogenic electron microscopy, like you mentioned, to see RAD52 interacting with DNA. And what they found was that it doesn't just form a simple ring like scientists thought it would.
Okay.
It actually makes a double ring structure.
A double ring.
Yeah, it's like two interconnected rings, almost like a tiny little spool. And each ring has 11 copies of the RAD52 protein.
Wow, that's pretty wild. And how does this double ring actually interact with the DNA?
Well, they found that it binds to this really important area called the DNA replication fork.
Okay, remind me what that is again.
So picture your DNA like a zipper, and it's being unzipped so it can be copied.
Mm-hmm.
The replication fork is the point where it's splitting.
Okay, I got it. It's like the point of action.
Exactly, and it's a really vulnerable spot. And get this, the double ring of RAD52 basically clamps onto all three arms of the replication fork.
Oh, so it's not just a little touch. It's like a full-on grip.
Yeah, it's a full embrace. And that's what's so remarkable about it.
So what's RAD52 doing there with that double ring? What's its job?
It's basically stabilizing the replication fork and protecting it from breaking down too much.
Okay, so it's like bodyguard for the DNA.
In a way, yeah, especially in these cancer cells that already have problems with their main DNA repair systems. When the replication fork gets messed up, which happens a lot in cells that are dividing rapidly, RAD52 steps in with this double ring and stops it from falling apart.
So that's how the cancer cell survives.
Yeah, it's able to keep growing even with all this genetic instability.
It's incredible that they were able to see all of this in such detail.
Right.
And not just with the cryo-EM, but also with this special single molecule microscopy that Dr. Spies' lab developed.
Yeah, they were able to watch these RAD52 DNA interactions happening in real time.
It's mind-blowing.
It is, and it shows that this protective role isn't just static, it's dynamic. There's all this movement between the protein and the DNA.
So we've got this unexpected double ring structure that's basically a lifeline for these cancer cells.
Mm-hmm.
That's a big deal, just understanding that. But how does this help patients? Like, how do we translate this into actual treatments?
This is where it gets really exciting because this structure is like a chink in the armor. It gives us clues about how to attack RAD52.
Okay, I like that analogy. Tell me more.
Now that we know exactly how RAD52 interacts with DNA through this double ring, we can start looking for drugs that can disrupt that interaction.
So it's like finding the weak spot.
Exactly. Imagine a drug molecule that fits perfectly into a groove on the RAD52 ring.
Okay.
And it blocks it from clamping onto the DNA.
That'd be huge.
It would. And the really encouraging thing is that RAD52 doesn't seem to be essential for healthy human cells.
Wait, really?
Yeah, that's what the research suggests.
So that means we could potentially target it without hurting healthy cells.
That's the hope. It means fewer side effects. And that's a game changer.
It would be amazing to have cancer treatments without all the horrible side effects.
Right. It'd be a whole new world for patients.
So are we close to having these RAD52 targeting drugs?
Well, not quite yet. Dr. Spies' team has found some small molecules that can bind to and inhibit RAD52.
Okay.
But that's just the first step. They need to be refined and modified to make them more effective and more specific.
And safe for humans, right?
Of course. And that takes a lot of time and research. They'll need to test them in cell cultures and animal models before they can even think about human trials.
So it's still early days.
It is, but it's a really promising start.
And it sounds like this research was a real team effort. They had structural biologists, biophysicists, people doing computational studies, cell-based assays, all sorts of imaging techniques.
Yeah, it was a huge collaboration. They had Dr. Picheri's group in Rome working with Dr. Ashley Spies' team at the University of Iowa.
It's amazing what they can accomplish when they all work together.
I know, right? And you need that kind of multidisciplinary approach for something this complex.
Absolutely. So if we had to sum up the biggest finding of this research, it seems like it's this double ring structure.
Definitely.
And how important it is for keeping these cancer cells alive. They even called RAD52 a DNA replication gatekeeper.
It's a perfect name for it because that's exactly what it does.
Yeah, can you explain that a little more?
So the two-ring structure lets RAD52 control what happens at the DNA replication fork when it gets stuck or damaged.
Mm-hmm.
By clamping onto it, it stops the fork from collapsing, and that allows the cancer cell to keep growing.
So if we can figure out how to jam that gate, we could potentially stop these cancer cells in their tracks.
Exactly, and we could do it in a way that's really targeted to the cancer cells that need RAD52 to survive.
This has been such an incredible deep dive. It's amazing to see this kind of cutting-edge science happening.
I agree.
So just to recap for everyone listening, we now know about this surprising double-ring structure of RAD52 and how it protects damaged DNA in certain cancer cells. Right? And that discovery could lead to brand new anti-cancer drugs that are more precise and maybe even have fewer side effects.
I'm really excited to see where this research goes.
Me too. And it makes you think about all the other proteins out there that we might not even realize are important. Like, what if there are other seemingly dispensable proteins that are actually crucial for other diseases?
That's a great point.
It's like there's this whole hidden world of potential drug targets just waiting to be discovered.
It's a whole new frontier.
It really is.
It's pretty inspiring.
It is. I mean, who knows what other breakthroughs are out there.
And what other diseases we might be able to target more effectively.
It's definitely something to think about.
Absolutely. It gives me hope.
Me too. It's a good reminder that science is always moving forward, and there's always hope for new and better treatments.
I couldn't agree more.
Well, this has been a fascinating conversation. I've learned so much.
Me too. Thanks for having me.
Thanks for being here. And to everyone listening, thanks for joining us on this deep dive into the world of RAD52.
It's been a pleasure.
Until next time, stay curious.